Namaskar. Today we'll study the thyrotrophin receptor. This thyrotrophin receptor, how exactly it functions, that we will study today. Thyrotrophin receptor is a G protein coupled receptor, and this G protein coupled receptor is a type of G protein coupled receptor, and the effector of this G protein coupled receptor is adenylate cyclase, which converts ATP to cyclic AMP. And the ligand for this receptor is thyroid stimulating hormone. So when this thyroid stimulating hormone acts on the thyrotrophin receptor, there will be production of the thyroid hormones T3 and T4. So you can refer to our earlier classes on thyroid glands, hyperthyroidism and hypothyroidism, and drugs for them. And just to remind you, this is a thyroid gland. We have the follicular cells here, and on these follicular cells we have the thyrotrophin receptor. TSH acts on these receptors, and from these follicular cells there is release of the thyroid hormones T3 and T4. And so you can see, so this is the follicular cell and this is follicular lumen. So this is your follicular lumen, this is follicular cell, and this is the blood capillaries. So if we plot this here in this figure, so this thyroglobulin is a protein with tyrosine residues around it, and we get iodide from diet. It comes to our plasma, and this iodide is uptaken, and by the action of thyroperoxidase in presence of H2O2, it is converted to iodine, and then this iodine is iodinated on these tyrosine radicals. These tyrosine residues are also converted to tyrosine radicals by means of thyroperoxidase. You can see this is the tyrosine, and tyrosine amino acid in presence of thyroperoxidase and H2O2, it becomes tyrosine radical. One hydrogen is lost here, so it becomes tyrosine radical. And iodide is also converted to iodine in presence of thyroperoxidase and H2O2. Then this tyrosine radical is iodinated to monoiodotyrosine. Then in presence of another iodine becomes diiodotyrosine. Diiodotyrosine in presence of monoiodotyrosine becomes triiodothyronine, and diiodotyrosine with another diiodotyrosine becomes thyroxine T4, T3 and T4, and these T3 and T4 are synthesized in the lumen. Then it is endocytosed by the lysozymic action. T3, T4 are released into the blood, and monoiodotyrosine, diiodotyrosine becomes the source of iodide again. So thyrotrophin or thyroid stimulating hormone TSH acts on a G protein coupled receptor, thyrotrophin receptor. And let us see the G protein coupled receptor. You can refer to our earlier classes on G protein coupled receptor. So you can see the G protein coupled receptor, there is G protein alpha, beta and gamma, and GDP is linked to alpha, and in the resting state, when the ligand binds to the receptor, this alpha GDP, there will be GTP exchange, and alpha, it becomes alpha GTP, and it gets separated from beta gamma as well as the receptor. And now this alpha GTP or beta gamma binds to the specific effectors and show their action, and this effector may be the enzyme or ion channel. Enzyme may be adenylate cyclase, phospholipase, ion channel may be potassium or calcium. So for the thyrotrophin receptor, the effector is enzyme adenylate cyclase. So there can be four classes of G proteins, and one first class family one is GS family, GS subfamily, and G olfactory. So GS subfamily, the function is activates adenylate cyclase. So when there will be binding of TSH with the thyrotrophin receptor, there will be activation of the alpha SGTP, and this alpha SGTP will bind to adenylate cyclase. There will be formation of cyclic AMP from ATP. So this cyclic AMP activates the protein kinase A. So how it does this? Protein kinase A has 4 subunits, 2 regulatory subunits and 2 catalytic subunits. And when cyclic AMP binds, so 4 molecules of cyclic AMP now will bind to the regulatory subunits, and there will be release of the catalytic subunits, which are active catalytic subunits. And these active catalytic subunits are the active protein kinase A. So protein kinase A, when without cyclic AMP, is inactive. With cyclic AMP, there will be release of the active catalytic subunits, which is the active protein kinase A, which so when this active catalytic subunit is formed, that active catalytic subunit will enter into the nucleus, phosphorylate or activate cyclic AMP response element bound protein CREB, which will recruit the co-activators CREB binding protein CBP. Now the CREB CBP complex will bind to the cyclic AMP response element and regulate the gene transcription. So here the gene transcription will lead to the production of proteins, that is the hormones, thyroid hormone T3 and T4. So T3 and T4 synthesis and secretion will occur because of this gene transcription, because of binding of CREB and CBP complex with the cyclic AMP response elements. So in this way, thyroid stimulating hormone, by acting on the GS type G protein coupled receptor, thyrotrophin receptor, activates the adenylate cyclase, forms cyclic AMP, which activates the protein kinase A, where the activation will lead to formation of the active catalytic subunits, which will enter into the nucleus and will regulate the gene transcription through the CREB and CBP complex. This is all about thyrotrophin receptor. Thank you.